Yeah, good morning, children. We are going to start with a chapter called photosynthesis in a series of chapters. What we are going to discuss here. This is an effort to make you understand and revise this topic more faster. So I'm not going to write much on the board. The complete 30 minutes we are going to discuss very, very thoroughly all this entire chapter. More importantly, you have to concentrate everything, all 30 minutes to what I'm talking here. So you need to lend your 30 minutes of today to me for this. In this 30 minutes, we'll be completing this chapter thoroughly. Without wasting any time, we'll start with this chapter called photosynthesis. The area where you normally are weak in photosynthesis is the last part called factors. First, I complete that and come back to the first part. The first important factor is water. Look at water. Water doesn't have any direct role in photosynthesis. In the presence of water, the cells are going to elongate. When the cells elongate, the surface area will be more absorption of CO2, more absorption of light, more photosynthesis. Next, water also have a role in turgidity of guard cells. When the guard cells become turgid, the stroma opens. When the stroma opens, more amount of CO2 can enter inside, more photosynthesis. Children, what did you observe? You observe that the role of water has no direct impact on photosynthesis, but will definitely impact photosynthesis indirectly, but by having a role on the plant. Finished. Next is temperature. You know pretty well that enzymes are thermolabile, means sensitive to temperature changes. Where are the enzymes involved? Both in light phase and dark phase enzymes are involved. In light phase, enzymes are relatively less involved. There are only two enzymes. One is FNR and ATP synthase. In the dark phase, every reaction is catalyzed by an enzyme. Therefore, between light phase and dark phase, where temperature has a role, the answer is both. Where the temperature has more important role, the answer for that is dark phase, because all the dark phase reactions are temperature dependent. Last point. C3 plants and C4 plants, you studied. Your teacher told C4 plants example is sugarcane, maize and sorghum. Sugarcane, maize and sorghum are seen in India, not in America. India is a tropical country. Temperature here is more. Easy way to remember. In C4 plants which are tropical, the temperature optima is more. At 40 degrees, they show maximum photosynthesis, whereas C3 plants, the temperature optima is less because they are temperate plants. So next time, you're not going to get confused here. Next factor we are going to talk about is carbon dioxide. Listen carefully. 350 ppm. The carbon dioxide concentration is around 350 ppm in the atmosphere. 350 ppm in the atmosphere. Your textbook says 0.03 ppm. This is the concentration of CO2 in the atmosphere. Listen carefully. In a C3 plant, all right, the saturation is reached at 450 ppm. In a C4 plant, saturation is reached at 350 ppm. What does it mean? In C4 plant at 350 ppm, maximum photosynthesis is seen. Atmosphere is 350 ppm. It means that the present level of CO2 is enough to reach maximum photosynthesis in a C4 plant. What about a C3 plant? The atmosphere contains 350 ppm, the saturation is at 450 ppm, means the present level of CO2 is not sufficient. Therefore, the amount of CO2 concentration you need to increase. That is the reason we pick a room like this, enhance CO2 concentration here. Tomato and bell pepper are grown in enriched CO2 concentration, where we increase the CO2 concentration from 350 to 450 such that they can reach saturation. These are called greenhouse crops. That's about CO2, children. The next important factor, and most importantly, CO2 is the most common limiting factor because the atmosphere is only 0.03. The majority of plants are C3 plants. Therefore, the present level of CO2 is acting as a limiting factor for a C3 plant. The next important factor is light. Look here, with increase in light intensity, the photosynthetic rate is increasing. As long as the light intensity is less, but beyond a certain point, when the light intensity is more, increase in light intensity does not have any effect on the rate of photosynthesis. Why like that? The other factors become suboptimum. The other factors become limiting. Law of limiting factors was proposed by Blackman. He says that whenever any factor, this is optimum. Whenever any factor is suboptimum, below optimum, at this point, the light will act as a limiting factor. When it is here, no. Why? Because the light is optimum. So suboptimum factor, any factor less than what is needed, maximum needed. that is acting as a limiting factor. It's very clear that light acts as a limiting factor in majority of CO2 normally acts as limiting factor. Light never acts as a limiting factor in majority of plants except for shade plants. Plants growing in a dense forest where the amount of light reaching is there is less. 10% of light is enough to show maximum photosynthesis. Therefore, light rarely acts as a limiting factor. And remember, law of limiting factors was proposed by Blackman. That point is equally important. The meaning of limiting factor is a suboptimum factor. Next. Shalom, now I go to the first part of photosynthesis. What I'm going to discuss here. What is the role of photosynthesis? 1. Photosynthesis, both light phase and dark phase occur during daytime. 2. Light energy is converted to chemical energy. 3. The role of photosynthesis is to A. Produce food, B. To evolve oxygen. 4. There are 3 kingdoms in Whittaker's classification in which photosynthesis is seen: Monera, Protista, and also what is called Plantia. 5. There are 2 types of photosynthesis: oxygenic and anoxygenic. Oxygenic photosynthesis is very common. Anoxygenic photosynthesis is seen in green sulfur bacteria and purple green sulfur bacteria. Von Neal reported that it is H2S which get oxidized, not water. Therefore, there is evolution of sulfur, not oxygen. 6. Photosynthesis is an anabolic process. 7. The cell organelle responsible for photosynthesis is chloroplast. After talking about this, remember, now we talk about the 2 experiments. Variegated leaf experiment, and the second type of experiment is Moll's half-leaf experiment. Half of the leaf is inside a test tube with KOH, half of the leaf is outside. KOH absorb all the CO2. The part of the leaf inside the test tube will not get CO2, no starch. It doesn't respond positively to iodine test. That is the role of CO2, Moll's half-leaf, don't forget that. Coming to variegated leaf experiment, half of the leaf I'm going to surround with a black paper. Then half of the leaf is exposed to sunlight. The part of the leaf which is surrounded by black paper will not produce any glucose. And the leaf which is exposed to light produce glucose. What does it mean? It not only explains the role of light, but also the role of green tissue responsible for photosynthesis. That is called variegated leaf experiment. It is the role of chlorophyll in performing photosynthesis in the presence of light. Remember that. Chalo children, now we are going to talk about the scientists' names. Listen carefully. Priestley, he took a mouse and he took a candle. He put it in a bell jar. Both of them disappointed. Candle was put off, mouse died because there is no oxygen. Then he took a bell jar this time. He took a mouse and candle. Along with it, he also put a mint plant under this. That made a huge difference. Everybody was happy, including Priestley. Why like that? Mint plant was taking CO2. Released by mouse and candle and evolving oxygen, and the oxygen was taken by mouse for its respiration and candle for its combustion. All right, so therefore all the 3 things were happy. Then Priestley wrote in his diary, all right, of course he is an architect of discovering oxygen. He wrote in his diary, he says that plants take bad air, means CO2, and liberate good air, that is oxygen. That is very, very important. That is about Priestley. Ingenhousz repeated the same experiment done by Priestley outside his house. Therefore, his name is Ingenhousz. In the presence of sunlight, the role of sunlight in photosynthesis was reported by Ingenhousz. Remember that. Aquatic plant outside was able to evolve oxygen bubbles in its pond during daytime, not in nighttime. Means oxygen is evolved only during photosynthesis in light phase. So only during daytime. Photosynthesis occurred during daytime. He reported that. Sachs for the first time reported starch is the end product of photosynthesis. SNS, Sachs, starch, easy way to remember. Next, the scientist's name is Angelman. Now listen here. I'm not going to take much time. Angelman, G-Y-O-R. What did he observe? He observed that there is more amount of light absorbed in blue and red. All right, blue and red wavelengths are absorbed by plants in large quantities. Green is the wavelength of light which is absorbed in small quantities. I'm sorry, this was not explained by Angelman. This is called absorption spectrum. In absorption spectrum, it is clearly reported that blue and red photosynthetic rate is more, whereas in green it is less. This is called absorption spectrum. Angelman, just like Superman, Batman, He-Man, he is associated with action spectrum. Now, what is the meaning of action spectrum? This is action spectrum. The rate of photosynthesis reported by Angelman is in fact more in blue and red. Rate. Angelman reported rate of photosynthesis, not absorption. Rate of photosynthesis is more in blue and red. Clearly it says that whichever wavelength of light chlorophyll absorb, the same wavelength the photosynthetic rate is also more. Therefore, he said that chlorophyll A is a chief pigment, because whichever wavelength of light chlorophyll A absorbs in large quantities, blue and red, at the same wavelength, the rate of photosynthesis is more. At the same time, Angelman said that, look here, it is not exact one-to-one overlap. Which one? Absorption spectrum of chlorophyll A and action spectrum are not exact one-to-one overlap. What does it suggest? It suggests that there are other pigments also which have a role in photosynthesis. That is also one of the inference made by the graphs which are there in your NCERT book. Next time, should not get confused. Angelman is associated with action spectrum. That's a very important point to note. Then we have got photosynthesis. Von Neil, I just now told you, anoxygenic, oxygenic. In anoxygenic, H2S get oxidized. In oxygenic, H2O get oxidized. In H2S, if it gets oxidized, there is no evolution of oxygen. If H2O get oxidized, there is evolution of oxygen. It means that whenever water is there, oxygen is evolved. Wherever water is not there, oxygen is not evolved. CO2 is common for both. It means that it is not the CO2 which is the source of oxygen liberated. If CO2 is the source of oxygen liberated, oxygen would have been liberated in both the cases, in the presence of H2O and also H2S. So the fact of the matter is, in the presence of H2O, oxygen is liberated. In the presence of H2S, oxygen is not liberated. Clearly suggests the role of oxygen, the role of water in the evolution of oxygen. Green sulfur bacteria, purple green sulfur bacteria, anoxygenic photosynthesis. Von Neil, you remember all this. And please also remember, cyanobacteria are the only blue-green alga which does not show anoxygenic photosynthesis. Their H2O get oxidized, therefore there is evolution of oxygen. So the bacteria with anoxygenic is green sulfur and purple green sulfur, and the bacteria with oxygenic, just like higher plants, is cyanobacteria, blue-green alga. Also remember, in blue-green alga, heterocyst, that word is important, spirulina, SCP, that is important, and nostoc and anabina are biofertilizers, even that is also important. Chalo, what are the color of chlorophyll A? Blue-green. What are the color of chlorophyll B? Yellow-green. What are the color of xanthophylls? All right, yellow. Old and mature leaves, yellow. What are the color of carotenoids? Yellow-red or yellow-orange. You remember these colors, A, B, xanthophylls and this. Now, what we're talking about here. Now I talk about light phase. This is called stroma, this is called membrane, and this is called lumen. Stroma, membrane, and lumen. Now listen carefully. On the thylakoid membrane, all of you know that in the chloroplast covered by two membranes, the proteinaceous fluid is called stroma, which contains circular DNA, 70S ribosomes, and all three types of RNA in which protein synthesis occur. Stroma is also a place where dark phase enzymes are present. All the thylakoids group together to form grana, that is called grana lamellae. The two adjacent grana lamellae is also covered by a thylakoid, and that is called stroma lamellae. Why? Because on either side of this there is stroma. So grana lamellae and stroma lamellae represent the membrane system, and the stroma represent the protein fluid in which the dark phase occur. Grana lamellae and stroma lamellae, light phase occur, and in stroma, dark phase occur. So let us now talk about grana lamellae. All the membranes are grouping together to form grana lamellae. Now this is a P680, which is a chlorophyll A molecule. Listen carefully. Surrounding it, you see the other photosynthetic pigments. You are getting confused time and again here, children. I want all of you to remember this. This entire concept is a photosystem or a pigment system. What does a photosystem contain? Four things. One, a reaction center, which is always a chlorophyll A molecule. Two, antenna complex or light harvesting complex, which include all the pigments, including chlorophyll A. Chlorophyll A, chlorophyll B, carotenoids and xanthophylls. That forms a light harvesting complex or antenna complex. 3, it contains the primary electron acceptor. 4, it also contains proteins to which this photosystem is attached. All this represents what is called a photosystem. Reaction center, light harvesting complex or antenna complex, which include all pigments including chlorophyll A, primary electron acceptor and proteins, all that represent a photosystem. All right. Now, remember, what is the chief pigment? Chlorophyll A. What are accessory pigments? Chlorophyll B, carotenoids and xanthophylls. Why you call them as accessory? Because they are not a part of reaction center. Chlorophyll A is a part of reaction center and light harvesting complex. That is the reason it is chief. Whereas the other pigments, B, carotenoids and xanthophylls, is a part of only light harvesting complex, not reaction center. Therefore, they are accessory pigments. In the presence of light, electrons get excited. Now it becomes P680 star. Easy way to remember. This is uphill. From P680 star, the 2 electron goes to a compound called pheophytin. This is also uphill. This is entire thing is uphill. From P680 to pheophytin, primary electron acceptor, it is uphill. Once it loses 2 electrons, P680 star now becomes P680 plus, naturally, because it is deficient of 2 electrons. This strong P680 plus, strong desire of wants to get reduced, means it wants 2 electrons very badly. This is going to break water into 2 electrons, 2 protons and half O2. 2 electrons, 2 protons and half O2. Now P680 plus gets these 2 electrons such that it again becomes P680. So easy to remember. Alright, so if somebody asks you which photosystem is responsible for breakdown of water, the answer is PS2. This is photosystem 2. This is responsible. If somebody asks you where photolysis of water occur, on the lumen side, where is evolution of oxygen, on the lumen side, then what? From pheophytin, the 2 electron goes to a compound called plastoquinone. Plastoquinone is not only electron acceptor, children, but also a proton acceptor. It accepts protons from stroma. Electrons from pheophytin, protons from stroma. Common sense, 2 electrons, 2 protons. PQ now becomes PQH2, plastoquinol. Plastoquinone is mobile. So it is going to move from the stroma side of the membrane to the lumen side of the membrane and releases the 2 protons and releases the 2 electrons to a compound called cytochrome B6F. Listen carefully. Pheophytin accepts 2 electrons and 2 protons to form PQH2. PQH2 loses 2 protons and 2 electrons again to form PQ. If somebody asks you who is the proton carrier from stroma to lumen, your answer should be PQ. If somebody asks you who is the proton pump, the answer is cytochrome B6F. You got it out. Why it is a proton pump? Because it is pulling 2 electrons from here. By pulling 2 electrons, it is allowing the loss of 2 protons into the lumen. Therefore, the proton pump is this. Proton carrier is PQ. Understand that. Why we say that protons are moving from less to more? Naturally. Why? Because there are more protons added because of photolysis of water. Because there are more protons added because of photolysis of water, we say that protons are moving from less to more. Less to more is uphill. Is energy needed? Yes. Who is going to provide the energy? Common sense. The downhill movement of electrons will provide the energy, and that energy will help in the uphill movement of protons via plastoquinone from stroma to lumen. Easy to understand. From here, the 2 electron goes to plastocyanin, which is a copper containing electron carrier, plastocyanin. This is uphill, children, up to pheophytin. From pheophytin to plastoquinone, plastoquinone to cytochrome B6F to a copper containing plastocyanin. This is downhill. Chalo, one more photosystem operate. This is P700, and this is the antenna complex, a light harvesting complex. It was discovered first, therefore it is rightly called PS1. It operates first, this discovered first. Remember the difference. Now, electrons get excited. Naturally, it becomes P700 star. Then it loses 2 electrons to ferredoxin reducing substance. When it loses 2 electrons to ferredoxin reducing substance, then it becomes P700 plus. Now the plastocyanin gives its 2 electrons to P700 plus such that it again becomes P700. From here, the 2 electron goes to ferredoxin reducing substance, ultimately to ferredoxin, and ultimately to a compound called NADP. Ferredoxin reducing substance to an iron sulfur protein called ferredoxin, and ultimately to NADP. What did you observe? This is uphill, this is downhill. From P700 to ferredoxin reducing substance, uphill. From ferredoxin reducing substance to NADP, downhill. You see 2 uphills and 2 downhills. What this entire process do? This downhill movement release energy that will allow the protons to get pumped from stroma to lumen. Now what did you observe, children? The last 2 minutes. There are more protons in the lumen, and there are very, very less protons in stroma. I'll tell you why there are more protons in the lumen. Reason number one, because there is photolysis of water contributing protons. Reason number two, plastoquinone is a proton carrier from stroma to lumen, carrying protons uphill from stroma to lumen. That is the reason lumen is a reservoir of more protons. Negative logarithm of H plus ion concentration is pH. When there are more protons here, the pH is naturally will be less in the lumen. Easy way to remember. Once we create a gradient, one fine morning, we allow these protons to pass through a coupling factor. This is called CF0 and this is called CF1. What did you study in photosynthesis and respiration? You studied that the movement of every two protons is going to release ATP. Why? Because protons are moving from high to low. When protons are moving from high to low, energy released. Here protons are moving from low to high, energy required. The downhill movement of electrons will provide that energy. Here the protons are moving from high to low, energy is liberated. For every two protons, one ATP. There are four protons are transported, therefore there are two ATP synthesized. This is called photophosphorylation. This is also called chemiosmosis. Why? Because you are breaking the proton gradient and producing ATP, therefore it is chemiosmosis. Did we produce ATP? Out of four protons, two protons are going to reach NADP. Two protons reach NADP to form NADPH2. Now I'm going to ask you a few questions. Who is the ultimate donor of electron? Water. Who is the ultimate acceptor of electrons? NADP. What are the products of light phase? NADPH2, ATP and half molecule of oxygen. Out of these three products, which are utilized in dark phase? ATP and NADPH2. Which is liberated to the atmosphere? Oxygen. Which occurs towards the lumen side? Breakdown of water, lumen side. Evolution of oxygen, lumen side. More proton accumulation, lumen side. Which are occurring toward the stroma side? Less proton concentration, stroma side. NADPH2 production, stroma side. ATP production, because the head of this coupling factor is there towards the stroma side. Synthesis of ATP, stroma side. Synthesis of NADPH2, stroma side. Less amount of protons in the stroma side. If somebody asks you, why less protons in stroma? Your answer should be A, plastoquinone is pumping the two protons, not allowing the protons to stay here. That is the reason protons are less. Reason number two, NADP is also accepting some protons and becoming NADPH2, making the stroma deficient of protons. If somebody asks you why there are more protons in the lumen, your answer should be because of photolysis of water and because of plastoquinone. So proton gradient is because of all this. So nice, I explained everything nicely. What happens when there is VIBGR up to 680 nanometers? Both the photosystems operate and this is called non-cyclic electron transport. What happens when you expose a photosystem to a wavelength greater than 680 nanometers? Greater than 680, this will not work. Alright, then this will be losing here to ferredoxin, reducing ferredoxin, and there is no. From ferredoxin, the 2 electron goes to plastoquinone. From ferredoxin, the 2 electrons. Again, for every 2 electrons, plastoquinone accepts 2 protons. The remaining story is known to you, PQH2. It loses 2 protons in this. From where we started here, we went like this and we went like this. Therefore, it is cyclic. Which photosystem will not operate? PS2. Why? Because you're exposing a photosystem to greater than 680 nanometers. Added to that, stroma lamellae, where the cyclic electron transport system occurs, doesn't have two things. It doesn't have PS2 and it doesn't have this enzyme called FNR, ferredoxin NADP reductase, which is responsible for NADP reduction. So if I am stroma lamellae, I don't have PS2, I don't have this 2 enzyme, why I should show non-cyclic? I'll pick cyclic. Imagine if I'm grana lamellae. Alright, normally I show non-cyclic, but when you expose to a wavelength greater than 680 nanometers, my PS2 will not work. Only PS1 work because PS2 maximize 680. If it is 681, only cyclic operate. If it is 670, both operate. If it is 570, both operate. If it is 370, both operate. Non-cyclic operate. These are the points which you have to remember with regard to light phase. Master O, why did you produce all this? All this is produced in order to convert carbon dioxide to glucose. Now all of you know that 6 molecules of RUBP, ribulose biphosphate. It combines with 6 molecules of carbon dioxide and form 12 molecules of phosphoglyceric acid. In the presence of 12 ATP and 12 NADPH2. It forms 12 molecules of phosphoglyceraldehyde. I convert that into 10 and 2. This 2 I convert them into glucose. This 10 I utilize in the regeneration of RUBP by utilizing 6 molecules of ATP. All of you know this. This is called carboxylation phase, which is a crucial phase. I'll tell you why it is crucial. This is called reduction phase because I'm adding hydrogens to the substrate. Addition of hydrogen is called reduction. Acids on reduction give rise to aldehydes. Therefore, it's reduction phase. This is called regeneration phase. I've regenerated RUBP. So, initial carbon dioxide acceptor, keto sugar, RUBP. Fast stable product, phosphoglyceric acid, 3 carbon compound. Hence, it is called C3 pathway. The enzyme responsible for this is Rubisco. This reaction is crucial. 12 PGA to 12 PGL. This is said to be reduction phase. ATP and ADP is utilized here. This is said to be regeneration phase in which I regenerate 6 molecules of RUBP. The total number of ATP utilized 12 plus 6, 18. Total number of NADP utilized 12. This is for how many Calvin cycles? 6. For 1 Calvin cycle, how many ATP? 3 ATP and 2 NADPH2. All these reactions occur in stroma of mesophyll cells if it is a C3 plant. Stroma of mesophyll cells if it is a C3 plant. This is called C3 cycle, also called Calvin cycle. Calvin is a scientist who worked on C14 and chlorella. He also reported that in photosynthesis, light phase is converted to dark, light energy is converted to chemical energy. That point is important. Now, what happens? Listen carefully. If RUBP combines with CO2, this is the 5 carbon, this is a 1 carbon, then you see the formation of 2 molecules of phosphoglyceric acid. This is called C3 cycle. Okay, no problem there. If the same RUBP is going to combine with oxygen, this is interesting, combine with oxygen, it is converted to phosphoglyceric acid and phosphoglycolic acid. Now this is phosphoglycolic. Listen carefully, we are in the last 1 minute of the class. RUBP, if it combines with CO2 and participate in C3 cycle, the number of PGA formed is 2. If the same RUBP combine with oxygen, the number of PGA formed is only 1 because this 5 carbon compound in the presence of oxygen break into 3 carbon and 2 carbon. What does it mean? This is called Calvin cycle, this is called photorespiration. What does it mean? There are more PGA formed in Calvin cycle, more the PGA, more is the PGL, more is the glucose. Here there is less PGA formed, less will be the PGL, less will be the glucose. Photorespiration is a wasteful process. If somebody asks you why this occur, this is because of the dual nature of an enzyme called Rubisco. When CO2 is more, it catalyzes this reaction. The same enzyme Rubisco, when oxygen is more, is going to catalyze this reaction. So you cannot change the attitude of Rubisco. You can't say that, come on Rubisco, do only this because that will be helpful. Don't do this, Rubisco won't change. So you have to create an environment where Rubisco has to only do this, not this. That is called C4 pathway. The last minute. All right, we take 6 molecules of phosphoenol pyruvic acid and we add 6 molecules of carbon dioxide and form 6 molecules of oxaloacetic acid. Children, this reaction occurs in mesophyll cells. And what happens to oxaloacetic acid? It forms 6 molecules of malic acid. What I do with malic acid? I take this malic acid into bundle sheet cells and convert them into 6 molecules of pyruvic acid. This is 4 carbon, this is 3 carbon, 6 4s are 24, 6 3s are 18, there is a loss of 6 CO2 accepted by 6 RUBP, the remaining story is known to you, 12 PGA, 12 PGL and all the reactions you know, this is called C3 cycle. What did you create? You have created an environment in SMS, sugarcane, maize and sorghum, where this Calvin cycle is allowing in bundle sheet cells. If it occurs in bundle sheet cells, what is the advantage? They are superized. Oxygen cannot reach there. RUBP can never combine with oxygen. When RUBP never combine with oxygen, no question of any photorespiration. But if you ask me, if they are superized, oxygen don't enter, no photorespiration, we understood. But even CO2 also will not enter because they are superized. There is a mechanism to increase CO2 concentration in bundle sheet cells. We take PEP in mesophyll, oxaloacetic acid mesophyll, malic acid mesophyll. It is transported through plasmodesmata. Superized walls will not hinder. Malic acid can easily enter into bundle sheath. Finished. When it forms pyruvic acid, look here, it is releasing CO2. So what we have done in bundle sheet cells is we created an environment in bundle sheet cells where there is always more CO2 thanks to malic acid, no oxygen thanks to suberized wall, such that there is only dash photosynthesis, no photorespiration. These are called C4 pathway, sugarcane, maize and sorghum. They are more productive, they are more advanced. They are tropical plants, no question of photorespiration. They also show Kranz anatomy, bundle sheets which are suberized in the form of many rings. So now you understood there are two alternative pathways, that is C3 cycle and C4 cycle. C3 cycle operate in tomato, bell pepper, Calvin, etc., where only this occur. But the problem with C3 cycle is there is a chance for photorespiration because Rubisco is also exposed to oxygen there. But in C4 pathway, Rubisco is never exposed to oxygen because bundle sheet cells are suberized, will not allow oxygen to enter inside. But CO2 is released here because malic acid is losing that CO2. Children, this tiny effort of mine will be extremely useful for you to understand this concept. If you think that I went very fast, again you listen one more time after reading your textbook. We'll meet again tomorrow with a great chapter. Thank you.